So I'd like to welcome you to the third session in this block of talks. So Nick Moore will be talking about bioinformatics and his CountESS project. Nick Moore has been a contributor to MicroPython and has spoken at several conferences before, including PyCon and LinuxConf AU. So please give a warm welcome to Nick.
Hi. Hi, everyone. Good day. My name's Nick Moore. I've previously talked at conferences like this about things like MicroPython and about Django and things like that. And now I'm currently working in bioinformatics. If you're considering changing around fields in this way, I recommend it. It's a great way to constantly feel imposter syndrome for your entire life. It's great. But it's a really interesting project, and I hope you'll find it a lot of fun, like I'm finding it a lot of fun. I'm going to start the talk by introducing some concepts about bioinformatics, just so you have some idea of what the hell I'm talking about. This work is funded by Walter and Eliza Hall Institute. And also I'd like to thank some of my collaborators from Brotman Batty Institute and the University of Washington Genome Sciences who are helping out, because I'm a software developer, not a scientist, certainly not a bioinformatician. So the only way to make a project like this work is to be constantly working hand in hand with people who actually do the science. Content warnings on this presentation: we briefly mention cancer, we briefly mention COVID-19. We do not go into any kind of gross medical details. This is about numbers, not about the actual diseases themselves. So bioinformatics in 9.5 minutes. This will be cool. We'll move through it fairly quickly. There'll be a chance to chat about this stuff over lunch or whatever. I've got the green say hi sticker, and I really mean it. If you want to talk about this stuff all lunchtime, we can talk about this stuff. Also, my colleague Alan is sitting right there, so you can bug him about bioinformatics things too. So approximately 9.5 minutes, and we're going to go through fairly approximately about bioinformatics. Starting with what the hell is bioinformatics? It's the analysis of biological data. So we get lots and lots of data from biological processes. How do we analyze them? Arguably, it got started in about 1952 when we first realized you could sequence proteins. You could break proteins down to their building blocks and sequence them. And so Albert Sanger sequenced insulin, so we actually properly understood how insulin worked in the body, et cetera, et cetera. And Alan Turing, of all people, decided to take some time off for inventing computing to also invent bioinformatics just in his spare time. In a paper that kind of derived the chemical basis of morphogenesis, before anyone knew how any of the actual parts worked, he'd already written a paper that turned out to be largely correct once we found out that we could read the parts. It's a pretty amazing work. It really got computing once computers got invented to do a lot of the hard work for us. Margaret Dayhoff was a very early pioneer of this stuff, writing software in Fortran to actually align proteins, sort of subsequences, and work out how they would all fit together. And really interestingly, this protein information resource thing is a very early free online database, 1984, and people are already going, hey, information wants to be free, especially if it's a health information. It's really useful information for researchers. I think that's an amazing thing. Perhaps unsurprisingly, given how close they are in our time of science, there's a lot of parallels between biological sciences and computer science, and I'll talk a lot about that here because a lot of you are software people. Cell biology. There's a lot we still don't know about cell biology, even though we're made of cells. That's mostly because it happens inside the cells, and what happens inside the cells stays inside the cells. Once you pull them apart to find out how they work, they're generally dead and not so interesting anymore. But experiments let us make theories about how they must work internally. So you can change the inputs, you can see what the cell does, generally dies, but you can measure that and make some conclusions about how the cell must be working. It's a lot like black box testing of software, where you don't need to look inside the box. You can still test how it's behaving just by changing the inputs and monitoring the outputs. The Human Genome, which I'm sure is a term you've heard, it's the program that all of your cells run. All of them run the same program. It's a bit like having lots of things running the same container image, but different parts of the code get run depending on what that particular cell is doing. There's 23 chromosomes in there, give or take. Each of them is a very long molecule, like a long tape of 100 million or so building blocks called nucleotides. There's 4 of those, so they make up an enormous long tape. Eerily like a Turing machine when it comes down to it. All of your cells, I said that already, all of your cells have the same genome. How it's expressed depends on what the cell's job is. These days, we can actually read the DNA. There's a technique called Nanopore Electrophoresis, which basically sucks the DNA molecule through a teeny tiny hole, a lot like a small child eating spaghetti in a disgusting way. And it reads the molecule out one base at a time. And so we can actually read the binary. This is pretty exciting. And using some other techniques that you might have heard of, things like CRISPR and things like that, we can actually write the binary. We can hack little changes into the binaries, which is way cool. So we can read and write the binaries. So of course we understand everything. And not so much. A bit like in software, reading and writing the binaries is great, but it's not the only thing. We do know a bit about the syntax of the genome. Genes code for proteins. They're wrapped in a kind of a header and a footer. This is getting eerie, isn't it? That sort of regulate how much they get run and things like that. Some of them are tiny. They have one little protein, hundreds of base pairs in the binary. Other one of them's huge. They have dozens of proteins, millions of bases. They're like those enormous classes you see in some bits of software. And we know that they're sort of wrapped up in this way within the chromosomes. There are no debug symbols and no comments. It's very inconvenient. What we can do is we can look at the genes and we can go, we think we know what this does, we'll give it a name. We can just assign them a name. It's very much like what you do if you're ever reverse engineering a binary. You go through with a pencil if you're very old, or with Deirdre if you're rather younger, and you assign a thing and you go, this function I think does this, so I'll give it a name. We know of about 20,000 genes that encode proteins. We think we know what they do. That totals about 3% of the entire genome. We know from other stuff, there's a reference in my things which I'll send you later, that 80% of it at least does something. So there's a fair way to go, I think it's fair to say. To say we completely understand the genome, well, no. People have said that. We can download the whole thing, we don't necessarily understand the whole thing. Okay, so I'm about to talk a lot about gene variants, so let's just talk about what that is. Those little symbols on the tape are called nucleotides or bases. And they vary. Everyone's genome is different. There's this concept of a reference genome, which is like what is normal. No one has that actual genome. That's just like a reference. It's a typical genome. At one point, one person did have that genome, I think, which was the guy who started the Human Genome project. Wasn't it his personal sequence? No? Anyway. So everyone's different in some way or another. The most common kind of variant is just like a single base change somewhere there, flip a bit on and off. If it's a really common thing that happens in people, we call it a polymorphism because it's many shapes. If it's rare, we call it a variant because it's a little bit more scary. Some of these variants are really easily observable. Infant lactose intolerance and adult lactase persistence are both variants in LCT and MCM6 genes. Infant lactose intolerance we'd call a variant because it's rare and it's scary. Adult lactase persistence we call a polymorphism because a lot of people have that. That's where you can still drink milk as an adult. Or hair colour is another example of a gene variant that we kind of understand really well. Well enough that we can look at the human genome recovered from a 5,700-year-old bit of chewing gum and tell what colour hair the woman who chewed that gum had, as well as her skin colour, eye colour, and also she had duck and I think it was chestnuts for lunch. Because we can read all that DNA out of a piece of 5,700-year-old chewing gum. We've also found correlations between people with severe COVID symptoms and long COVID, and a particular variant on FOXP4, which is another gene that seems to be involved in lung stuff. So that's really interesting and might lead to some understanding of how COVID works and how to help people with long COVID. That's a correlation they found. We don't actually know necessarily why yet. There are more subtle problems as well. In cancers, cells are damaged by radiation or chemicals or just bad luck. Cell repair mechanisms then can either fix that damage or kill the cell off if it's unfixable. Those cell repair mechanisms have genes. They're created by genes. Those genes can have variants, and some of those variants can reduce the effectiveness of those repair mechanisms. It's much harder to find these variants, or how important they are, because these are rare things and people's lives are complicated. And it's a little bit like looking for a bug in an exception handler in live code that's already exposed to the internet. It's very hard to simulate this sort of stuff in the real world. Which brings me to multiplex assays of variant effect. And that's my rodent of unusual size, which is what the thing always reminds me of. There's a few thousand known variants of these sorts of cellular repair genes, and there's a lot more we don't know about because we've never seen them clinically. They've never happened in a patient who happened to have a particular cancer of a type that saw them go to the hospital and have their genes sequenced and so on and so forth. We haven't seen them clinically. Some of them might be serious. A lot of them are going to be just background noise, so they're not that important. But we want to know which ones are which. So multiplex assays take a different approach. They say, well, let's make all the possible variants. Let's get those machines that can read and write the binary, like I was mentioning before, and we'll write all the possible variants, and we'll make an experiment on all the possible variants, and we'll measure we'll sort of score them. How dangerous is this, basically? The outcome is the same. We want to know which variants you should get worried about and which variants you shouldn't, because that way you can tailor treatments to people's individual genetics. There's a resource that Alan talked about on Friday called MaveDB that exists to store and kind of retrieve these sort of result sets so that people can compare them and use them and so on and so forth. So look that one up on video if you weren't there. I'll talk briefly about a couple of techniques that are used for these because they're important to the software we're developing. One of them is a thing called saturation genome editing. This particular paper studies about 4,000 single base variants of BRCA1, looks at the cell growth assessed after 11 days, I think it is, and has it basically tries all these different possible variants and assesses how much they have affected the cells. It has good correlation with the variants we know of clinically, so it looks like it's really valuable work, and it identifies some new pathogenic variants, some new dangerous variants, so that could be really, really important for future medicine, for treatment. If you happen to have one of those variants, that might be very important to your life. This shows a little chart of the outcome of that study where you can see that a huge number of these variants on BRCA1 really, they have a score around 0. They didn't make much of a difference. There's a bunch more that have a very bad score. They made a big difference. BRCA1 no longer worked. Sorry, it says up there LOF, which means loss of function. The BRCA1 cellular repair mechanism doesn't work anymore. And there's a bunch of intermediate ones, which is really interesting. Those are things that it works still, but maybe not as well as it should. Similar work is underway on similar cellular repair mechanisms such as BRCA1 and PALB2, which are similar kind of mechanisms and also very important for cancer research. Another technique I just want to talk about briefly is a thing called VAMP-seq, which is really cool. I mostly want to talk about it because it takes jellyfish proteins and attaches them to a genome. Basically, you make cells of every possible variant of a particular gene. You attach a fluorescent marker protein to that gene as well, so it's expressed along with the thing that you're doing, and then you see how those cells reproduce. The more the gene is expressed, the more the cell glows, and then you actually physically sort the cells into bins based on how much they glow, and then you sequence each bin. Then you sequence each bin and you count the population of each sequence, and from that you can derive a score. The important bit about this is you can derive then a heat map of which variants where on the gene matter the most. It's a very cost-effective technique because instead of running thousands of separate sort of little tests. You're putting things in big bins at a time, which makes it a little bit more effective. Currently being used to investigate factor IX haemophilia, which is really interesting under various conditions. So that's a completely different thing. Now, you've probably noticed that the technique here is completely different to the technique I talked about before, SGE. We have different inputs, we have different outputs, we have different maths, scoring is different, etc. They're using some of the same tech, but there's some big differences. So the software I'm writing isn't about that stuff at all. The software I'm running is about counting things. Now, computers are really good at counting things. They've been really good at counting things ever since they got fed punch cards, and we had things called tabulating machines and so on. They're really great at counting things. So this is really quite a simple problem to solve, obviously. From the paper I've just mentioned on SGE, we have data for 13 exons over 163 files. We have 350 million sequences. We have 66 billion bases in those sequences, which isn't a ridiculous amount of data. I mean, it's a lot, but it's not ridiculous, right? But those data sizes are only, they're increasing, but it's not ridiculous as such. So obviously there's an easy solution to this. We just put them all in a text, we put them all in a text file and we run sort on them and we run that through unique and that spits out the counts. It's done, problem solved. It's already optimized, it runs on your computer, it's multi-core, it's fine. You've already got it. If you've got any kind of Unix machine, it'll work out fine. Unfortunately, the problem is humans. The actual problem isn't sorting and counting. The actual problem is file formats and metadata and filtering and grouping and transformation and statistics. You have to do all of these things to the numbers before and after you count them. Statistics is particularly important because you don't want to do this, where you find a correlation because you're looking for a correlation. For anyone who can't see it, this is a correlation of the Internet Explorer market share versus the murder rate in the United States of America. And you can see they're very nicely correlated if you graph them in the right way. You don't want that to happen. You need to run tests as you look at this data to make sure that you're seeing a real effect and not some kind of statistical correlation. Previously, a lot of this work was done in spreadsheets. There's some pretty major problems with that. For a start, we're heading up into the billions of rows, which doesn't play very well in Excel. Also, Excel has this real tendency to want everything to be a date, and genes have names, like I mentioned before, and some of those names are things like Sept2 and March 3, and Excel thinks they're dates. This paper estimates that roughly 20% of cell biology papers have errors introduced by these kinds of things. It's a bit out of date now, but it's a ridiculously large number. Also, just any series of digits with one E in them is turned into a floating point number. Thanks, real helpful Excel. So spreadsheets are not a great thing for this. So a lot of people write programs, which is great. Python, R, very popular. To coordinate all of that stuff, you probably need some bash script as well, and maybe a little bit of a make file. Yeah, it's fine. We can learn how to write some make, maybe snake make as well. We can coordinate all of these pieces together, and all you have to do is take someone who's a newly minted cancer researcher, keen to do cancer research, and send them off to learn Unix and Python and R and all of this stuff for like a year or so, because they have got nothing better to be doing with their lives. Actually, it would be a lot better if we could just get them working on cancer research straight away. So previously, we've had a tool called Enrich2, which is a GUI-based tool that people can feed these data files to, have them processed, they get spat back out again. It's very nice. It's GUI-based. It's easy to use. It's written in Python 2. It's specific to some specific experimental setups, though, and it's difficult to kind of grow from where Enrich is to cover all of these different kinds of experiment that people are coming up with, because files are always increasing. Scientists are always coming up with new kinds of experiment to run. That is part of the point of doing science, is you should be able to say, I've got a new way to do this. I want to try my new way of running this experiment. We want to enable that. So we've started on this new project called CountESS, Count-Based Experiment Scoring and Statistics. So as the name suggests, it's all about counting things, but it's also about the scoring and the statistics and how to support multiple kinds of experiment. It's got a graphical user interface because our users like to use programs that look like programs, not like scripts. But it's plugin based, so you can add and remove modules from it really easily. It uses flexible data pipelines, so the data kind of flows through it neatly. And rather than having a whole kind of bunch of, here's my program and my other program and my make file and calls this and calls that, it just has a single text configuration file that binds all that stuff together. Its architecture looks something like this, to try and keep all the modules a bit separate. There is a GUI. It's written in tkinter. I suspect it will not always be written in tkinter because that has proved not necessarily a great idea. And as per Russell's talk just before this, there are alternatives that are definitely worth considering, but it's very separate from the rest of the code, so I'm not actually all that worried about that. We can come back and replace that later. As an overview of what it currently looks like, here is a screenshot of what it currently looks like. The nice thing about tkinter, it does actually work nicely over X11 forwarding if you've got to do that. The window's split into 3 parts, basically. There's this tree of plugins and how they modularly connect together to flow data in and out of the system. On the left there, things are loading things up from files. On the right, they're writing things out to files, and the data kind of flows through all those modules on its way through. It's fairly drag and drop, state is saved to configuration files, etc. Each plugin has a configuration screen, so you can just fill in the blanks in the form, all that's saved to the config file. And most essentially, as you're doing this stuff, there's live data previews of stuff. Not the whole file, 100 million rows or so at the moment. Maybe we'll make that a little more flexible as we go along, but the idea is that it processes a bunch of rows so that you can see what you're doing. If you're writing a regular expression, you get to see that live, that you're making the changes and that it's working, which is great. There's also CLI, which you can run that just basically runs the same config file, but run it without having to look at it, which is useful if you want to use some bigger computer somewhere further away. Some of these runs to run the whole file can take hours and hours to run, so it's kind of nice if you can just SSH somewhere and do that. All right, the config files are really simple. They're that ini file format that you see everywhere because it's easy to diff. It works nicely in revision control. It's relatively simple. You can hand edit it if you have to. It's loaded and saved by the GUI. It's loaded by the CLI module. All the fun stuff happens in here. This is the pipeline code is basically all about coordinating multiple processes and the flow of data through the system and all this. All the tricky stuff happens in there. It's all hidden away nicely in there. It's a work in progress. It can be improved. There's lots of work to do in there, but that's what it's aiming to do. It calls out to the plugins and makes the plugins do things for it, and the plugins can be written in such a way that they don't need to think about multiprocessing. You write the plugin in a way that does like a map and a reduce, for example, and let the pipeline worry about how to make that actually happen. So there are basically 3 kinds. There's reading files, there's altering your data, and writing files back. They all subclass one of the many subclasses that are available that do all of that kind of structural stuff about this is a map reducey kind of plugin versus a very large number of the plugins literally just take a row at a time, transform it in some way, and spit out another column into that table. So if you want to calculate a score, for example, you might take a count and another count, and you might divide one by the other and take a log of that, and that will give you a score. You don't need to run that all in one place, all on one table or anything. It can run on many rows separately in different places, on many CPUs concurrently. So we allow that to happen by not making the plugin writer understand that. They can just implement the function that works on a row and not worry about anything else. So that's nice. Anyone can write a plugin. This is actually one of the coolest things in Python recently is this whole entry points mechanism. If you write a little clause into your plugin's pyproject file, all it needs to do is say, hey, this has an entry point. It's a CountESS-Plugins entry point. And you declare that basically, here's my plugin. When someone installs that with pip or whatever, CountESS can actually see that that plugin exists, and it can write it. The code in CountESS looks a little bit like that. It basically just goes through all the entry points from that import lib entry points function, and it says, are these valid plugins? Do they actually derive from that base plugin? If they do, then we'll count them, we'll make them available to the user. And lastly, the libraries underlying all this stuff, So a lot of this stuff runs on pandas because pandas is really handy for this, and NumPy. There are other libraries too. For example, there's one called Minimap2 that I've wrapped up to be a CountESS plugin. Minimap2 is an example of a thing that runs per row. It takes a variant and it turns it into a little kind of a diff almost. So you have a sequence and you say, I want to know where this sequence comes from and how it's different from the reference genome. So it can run once per row, and wrapping that up takes a dozen lines of code to just basically say, all right, for each line, this function gets called, so this function just needs to look up this particular variant and compare it and stuff like that. So that's really good. Just as an example, I've got 5 minutes, so this is perfect. As an example of CountESS used in Anger, this is kind of loosely based on that SGE work before. So the first, as you select the little plugins on the left pane there, it shows you their config and their own individual output on the right there. So the first thing is they're loading up all the FastQ files. 163 files get loaded. And what it's actually doing is grabbing the first little bit out of each file just so you have a representative sample of that data. And you can see that data down in the bottom sort of third of the screen over the right there. So that shows you the actual contents of those FastQ files. And FastQ is a particular file format used a lot in this thing. There's another plugin there that literally is just extracting some data from those columns that are in the FastQ file and altering it a little bit so that we can use it later. Along with those big FastQ files comes a little metadata file called SRA run table, and there's some metadata in there we need. So we also load that up. It's called SRA run table.txt. It's a CSV file. Who's surprised? So it basically reads us. There's a CSV reader that reads the CSV file. And then we have to transform that a bit because it's also not quite what we wanted. So we pull some different columns out of it that aren't actually CSV columns. They're just like embedded within the text strings in the CSV. And now we can do like a database join of these two data sources using a join plugin, which is one of the built-in plugins. We have variants. We want to compare them to the reference genome. So we have a separate file again with the reference genome for each of the exons in it. We then join that with the data. I think you're seeing where this is going. The data is all kind of flowing together. We split out data one way for the things that are unchanged. Oh no, I pressed a button. We split out the data one way for the stuff that's unchanged and one way for the stuff that is changed. Oh, I pressed the wrong button. Sorry about this. And then finally we call the variants. We say, okay, they're changed. These are all changed. In what way are they changed? So we go through and we make like a little diff for each one. And then we take counts of each diff. How likely are these, or how common are these compared to how common are the ones that are unchanged? We calculate a score from that. And finally, because this is science, we write it all out to a CSV file, because CSV files make the world go round. And all of that's in a configuration file. So you can change any one of those steps, and it's relatively easy to take someone else's work, replicate it, alter it a little bit, maybe just change the way it's scored, maybe just alter the way you filtered the data early on. Maybe you add in a step. You just go back there somewhere and say, I want to throw out all of these ones because these ones look like garbage to me. These low quality reads I'm going to get rid of, and I'm going to see whether my statistics come out nicer at the other end. The idea is that this should be very flexible, very easy for people to change. If you change your approach, you can still, like your sequencing approach, you could change the first few steps while leaving the rest of the steps alone and check that the answers still come out about the same. If you wanted to change the scoring, you can leave the first steps alone and change the last steps. You could change the middle if you happen to change the ways you're thinking about things. So that's CountESS. Hopefully we've seen a little bit about bioinformatics. We've seen some interesting problems that we want to solve and why they're important. One of the really lovely things about this job is at any time you feel like your work is pointless and you're staring into a video monitor all day and night for no reason, you see BRCA1 here and ovarian cancer there and so on and so forth, and you realize, oh, hell yeah, no, this is quite meaningful at the end of the day. And hopefully we can really help move people faster into those research activities. hopefully you've seen some interesting stuff about Python techniques, a little bit about the entry points and stuff, and maybe a nifty approach to data processing. Look, I think that this thing is actually useful beyond bioinformatics potentially. I think that there's potential for this kind of approach to work in other sciences as well, I hope. I'd love to talk to people about that if you're working in some other science as well. And I think it's been a really interesting project, and I'm very glad to be working on it at the moment. So thank you very much. I think we should have time for a couple of questions.
Thank you very much, Nick. Okay, we have time for one question. And if you have any additional questions, please ask Nick afterwards in the lunch hallway track. All right. Do we have any questions?
No? I should also mention, actually, just while I've got the mic here, that URL will give you all the slides and all the notes from all the slides. So if you wanted to see anything closer up, it's there.
Yes, we have one question.
So you're putting together a workflow for crunching lots of stuff.
Yep.
You mentioned briefly that you're spreading out the work. So do you have like a particular technique you're using, or pickling and sending the jobs around? How are you spreading out the work?
At the moment, it's all running on one computer rather than distributed. It could also be, the back end of this thing could also run on Dask, for example. It does have distributed sort of pandas, but distributed. And we've worked with that previously. I think that's probably a good way to go. At the moment, I've been concentrating on getting it working on sort of multiple CPUs and that abstraction layer of how do you write a plugin that knows enough about what it's going on that it can say, hey, I want to do a map reduce. Or, hey, I don't care, I just want to work on a row at a time. Just give me rows. I just want raw rows, and I'll give you back a column. What I want to do is get that abstraction right, and then in some ways that will take care of itself, because whatever backend you choose to run it on, it becomes its problem, right? How does it assign CPUs? Does it distribute stuff? In some circumstances, it doesn't need to. The other thing that I ran into very quickly, and if anyone follows me on Fediverse, you might have seen me whinging about this at one point. I finally got to the point where I was using all 16 CPU cores on my computer, and it was great, and it made a lot of loud noise with the fans all turning on and stuff, and I went, this is brilliant, and then I ran out of RAM, and it was killed by the OEM killer. And so that really, one of the really important bits is actually not just how do we use all the CPUs, but how do we not read all the data? How do we wait until some data comes out the other end before we read more data? And I think that's a worthy problem of evaluation, but also it's something that the plugins kind of, if we get the API for the plugins right, we can come back to it in some ways, because the plugins can then exploit that new understanding without changing the plugins or changing people's experiments. Like conceivably, we can already run a lot of these experiments on one computer in one place. I mentioned before that the research is underway on PAL-B2, and I spoke to the guy who's researching that, and I said, so have you got a similar amount of data to this BRCA1 one? He says, yeah, it's similar, it's 3 times as much. So yeah, literally in the space of a couple of years, data has tripled in size that we're dealing with. So yeah, I think it's quite likely that these problems will get bigger and bigger and bigger, and we'll just be chasing Moore's law the whole time. So, but it's a really interesting thing to consider: is do we use Dask, do we use Spark, do we use a million different possible clustering things?
Thank you, Nick. We'd like to offer you this gift as a
Thank you.
token of our appreciation for your interesting talk. Thank you very much.
No worries. Thank you very much. Thank you.